appreciate the opportunity to come visit and talk to old friends that I've known for a long time and talk about clinical sequencing, and it's been a really good visit so far. So I apologize, my title may seem somewhat vague, but as a trainee you're kind of roped into lots of projects, and so it's not like I can have like a 3-word title that summarizes the last 4 years of my life. So it will be kind of a collection of topics that I've worked on in the last 4 years. Just to give a quick overview of what I'm going to talk about. The first few sections of my talk will be focused on really clinical sequencing topics. I'm going to talk about the performance of some methods to detect cancer-associated mutations, particular internal tandem duplications in the FLT3 gene and AML samples, leukemia samples, and then the performance of different methods for detecting low-frequency variants in a specimen due to sample heterogeneity, tumor purity, and tumor heterogeneity. And those are both have been published, so I'm going to go over them fairly rapidly because they've been in published literature, and anybody who's done the work here is very familiar with the results of these sorts of studies because you learn them yourself. And then I'm going to move on to some work that I've been involved in more recently that's not published on contamination in NGS experiments. I've looked at different sources of contamination in multiplexed pooled sequencing, and then talk about some approaches that one might use in the clinical laboratory as sort of routine monitoring of contamination, which I think probably should be implemented in a quality assurance type program. And then finally, I'm going to talk about a recent publication from Tim Lay's group, which I've been involved in, on targeted sequencing of somatic mutations in AML samples to try to understand if subclonal cell populations have any sort of functional properties that may ultimately be clinically useful. So as Colin mentioned, I did my training here, and I left in 2010 to do lab medicine residency at WashU, and at that time, the clinical next-gen sequencing is what appears on the slide. There was nothing out there. However, about the time when I started residency, this paper came out from Mary Claire King and Tom Walsh, which when I went to the people at WashU to do clinical sequencing, this is the paper I had in my hand, because I think it was really the first example of clinical grade next-gen sequencing that detected a range of different variants that you observe in disease, in cancer, and inherited testing. And so the paradigm from this paper is really what we've implemented at WashU and what a lot of people in clinical labs have implemented, and it's shown here. I made this slide many years ago now, but it essentially outlines what Mary Claire and Tom Walsh did. You take DNA from some sample type, you fragment it, you make a next-gen sequencing library, you enrich for your targets of interest, you do pooled multiplex sequencing of multiple samples to gain efficiency, and then you do this at high depth of coverage. You map the reads to the genome, and then you use multiple informatic approaches to identify all the variants that you might expect in the sample. And so the key things I think that I learned from this paper was that you need high depth of coverage and you need multiple tools to detect variants. So that's kind of the paradigm that I thought was workable, and I think that's been borne out in the last several years. So at WashU, we started our initial panel in 2011. Didn't do much sequencing in 2011, but that's when we officially launched. And it's been through several iterations so far. We're now running this version 2, where we report 40 genes. We sequence more than that, but we just report 40. And then version 3 is in progress. So when we initially sort of implemented this assay, though, there were several important questions. And I'm going to talk about 2 of them that we addressed today. So the first is, can we reliably detect more than substitution variants? Mary Claire's paper showed that you could do that, but most of the action in the next generation sequencing field was in substitutions. And so it was unclear how well you could detect other mutation types. And obviously, more than just substitutions cause disease. And then the second question, which was unique to what we were trying to do compared to Mary Claire's paper, was the fact that in cancer, the variants are somatic mutations. They may be present at low frequency in the sample for many reasons, tumor purity, subclones, and that sort of thing. And it was really unclear how to be able to detect low frequency variants in a single sample. So that was a question that we really needed to address right away. My thinking was that this test should replace the existing tests. So we don't want to add it on. It should be able to do what existing tests do, and then potentially more. And most targeted testing of say KRAS mutations had reasonable sensitivity, so we needed to be able to at least do as well as those tests. So we set out to address the indel question with the FLT3 ITD mutation, because It's an important marker that people test for routinely in leukemia. And the insertions span a broad range, so we felt like if we could detect that range of insertions, then maybe that would be an indicator of how robust the technology would be to more general testing of other genes. And it was unclear what software was going to be the best for this. So this was kind of one of our first experiments. So we started out fairly modest sort of test set. We had 20 AMLs that had confirmed ITD insertions by PCR and capillary electrophoresis, and then we had 29 negative samples that were also confirmed by the same method. Included AMLs and some lung cancer specimens. We quickly found out that only certain methods could detect these insertions, and they took into account that there are that when you have an insertion, there's certain signals in the sequence data. And so I'm illustrating that here. I'm showing on the left just a cartoon alignment of paired end sequencing reads, and there are some reads where one mate in the pair is unmapped. So that it's mate mapped somewhere in the genome, but the other pair did not. There are other reads where both reads in the pair mapped, but part of one read was not fully aligned. And if you actually look at the sequence data, you can see this, and you can see evidence of these insertions. But you need a tool that uses those data to identify insertions. And there's only one commercially available tool at that point that did this, and it was Pindle. It uses a pattern growth method to take into account unmapped reads and partially aligned reads. And then I also thought that if you took all the reads at a locus, including the mates of mapped reads that were unmapped, and you assembled them together de novo, maybe you could reconstruct an insertion and identify it that way. So we found that those two methods were really the only ones of the commercially available ones that could detect the FLT3 insertions. And so we tested those on our 20 cases. Some of these were blinded, and we found that those two were the only ones that detected the ITDs. And in fact, in our set, it did detect an ITD in all of them. I'm showing that in green, the bottom two rows, Pindle and an assembly by Frapp. So these methods had 100% sensitivity for ITD positive cases. It detected all but one of the alleles that were in these cases. Some had more than one allele. And it had 100% specificity in this modest case of 29 samples. So this seemed to be, these approaches seemed to be the best for this particular case, and we implemented Pindle for our routine testing of cancer samples. So we also compared the allele fraction from PCR and capillary electrophoresis to an estimate of the allele burden from next-gen sequencing, which is somewhat tricky. And as you can see from this slide, the correlation isn't great. But it is remarkable to note that NGS detected even very low-frequency ITDs, in fact, down to about 1% abundance as judged by capillary electrophoresis. So this approach seemed to be fairly sensitive, have a good limit of detection. And so this kind of is lead into the second question: how well can you detect low-frequency variants of other types, such as S and Vs? And how much coverage do you need to reliably detect those variants? So to answer that question, we made some mixed samples in the laboratory, some synthetic mixes. And before that, we first sequenced Hapmap samples independently so we could identify a gold standard set of substitutions in these 5 samples. And then we did the mixes. We took one of the samples and mixed it with the 4 others at various proportions to create kind of a mixing study data set. And we sequenced them in pooled sequencing with our assay to high depth of coverage so we wouldn't be limited by sampling statistics, and applied standard tools, publicly available tools to identify the variants. And we used 4. The first 2, SAM tools and GATK, were well known and kind of the industry standards in the field, but they were really designed to detect constitutional variants, and they had not been applied to cancer testing at that point when we started this experiment. And so we didn't really know how those would perform, but since they're designed for inherited variants, which are present at either 50% or 100% allele fraction in the sample, it was unclear how well they would perform. We also included two other software packages, VARScan 2 and Splinter, which are not designed specifically for inherited variants. And so we would expect it to perform better, but we didn't know exactly what their performance would be. Splinter is a program that was written by somebody at WashU to detect low-frequency variants, and it requires spike-in controls to estimate the background error and that sort of thing. And so it's kind of a customized approach that required additional laboratory manipulations, which may detract for some applications. So when we ran these packages on our mixing study. We observed the following results. As expected, some of the tools that were designed for inherited variants did not perform very well. So SAM tools, for example, surprisingly, only detected about 50% of the variants, even at 25% variant allele fraction. So on the x-axis here, I'm showing the mixed proportion. And in the mixed proportion, a heterozygous variant in the minor population would be half the mixed proportion. So here, it'd be 25%, 10%, 5%, and 2.5%. And the y-axis, I'm showing the sensitivity. So SAM tools did not perform very well. GATK detected 100% of the variants at 25%, 97% at 10%, but then had a pretty steep cutoff at variants less than 10% VAF. The other programs performed significantly better, VARSCAN2 and Splinter, detecting more than 90% of the variants, even at 2.5% allele fraction. So the next question is whether there is any trade-off between the sensitivity and the specificity. And as you might expect, there was. So across the entire assay, the positive predictive value was still pretty good, 95% or higher, but VARSCAN2 did call a lot of false positives. So these are variants that you may want to review if they come up, and based on our gold standard set, were not in the initial samples that went into the mix. So, but the panel includes some non-coding sequence, and actually, if you just looked at the false positives in the coding sequence, there was only one coding false positive in the entire study. So, as far as variants that you'd actually report, the performance was pretty good. And in the paper that we published these results in, we actually found that if you filtered the reads before applying the tools, you could reduce the non-coding false positives down to only 1 or 2 per sample. So that's maybe a strategy that would be useful for routine testing. So we also wanted to determine what the coverage level was that was required for the optimal sensitivity. And so we took our set of mixed samples and computationally downsampled just by randomly sampling reads to achieve some mean coverage across the assay. And we determined that about 400x coverage observed at a position was required for maximal or near maximal sensitivity. So that's actually the coverage threshold that we use in our clinical test. If a certain number of positions don't meet 400x coverage, then we call it a limited study or a failed assay, and we have to repeat it. All right, so just to summarize that part of the talk, so FLT3 ITDs, which we think was a good starting set to determine the performance of indel detection and next-gen sequencing data, can be detected with the right tools. And then when you're trying to detect subclonal variants or variants in mixed samples, there is quite a bit of heterogeneity in the performance based on what program you use. And we found in our analysis that GATK has a pretty hard cutoff at 10%. VAF of VARSCAN2 detected down to about a 4% observed allele frequency. And obviously, because of sampling statistics, coverage depth is an important variable. And then, as I said, if you filter reads before you run these packages, then you can perhaps reduce the false positive rate.
Can I ask you a question now before you go on to the
Sure.
section? Can we go back one slide? So I guess I would have thought in the left panel that the lower frequency, like the 5 and 10%, would have been more affected by the low read numbers.
Yeah.
And not the opposite. Do you have an explanation?
I don't have a good explanation. So one thing is there's heterogeneity in the coverage across this assay, and so there could be it could be that these variants are in a region that has excellent coverage across the entire downsampled data set, and so it wasn't affected by the downsampling. That's one possibility. It is an interesting observation that that performance is pretty static across the coverage range. Yeah, and also this was one data set that was downsampled. So all the variants that are detected at the 10% level, there's probably the same variants. So maybe it's an artifact of the mixing that we did. So just to move on to the next part of the talk. So this is something I've been working on for the last couple of months. Next-gen sequencing experiments are complicated, multi-step protocols that involve lots of liquid handling. Often, in our testing, They do it in microtiter plates. There are samples that are right next to each other. Mixing can certainly happen. In the normal testing procedure, samples are marked with an index sequence, and then they're purposefully mixed together, and they have to be informatically separated. And this is done in our test at the sequencing stage, but there are laboratories that do it at before the capture hybridization stage. And that means that you only have to do one capture for 30 samples instead of 30 captures. So that can really increase the efficiency, but perhaps it can increase the level of contamination as well. And the other thing is, if you're sequencing cancer samples, you expect low frequency variance. So a little bit of noise at the bottom may actually be something real that you want to detect, and that can also be contamination. So being able to distinguish those two is perhaps a necessary exercise. And just to talk about one example of contamination. So this happened at an unnamed genome institute in St. Louis. We were sequencing 2 primary AML samples, and this was multiple samples from these 2 patients. I think there were at least a dozen samples in this one experiment. Each sample, so the dozen samples, were separately prepped for sequencing. The libraries were made, they were indexed, and then they were pooled together for a single capture, and then sequenced, and then computationally demultiplexed, and then analyzed. And so in this first figure, I'm showing the results from one sample. So I'm showing, these samples have been, they've been whole genome sequenced. We knew where the variants were, and we actually captured the somatic variants that we were interested in. And so on the first plot here, I'm showing each point is a somatic variant, and I'm showing them from the AML 43, which is one of the samples. This sample had over 1,000 somatic variants that we were interested in. And I'm showing the variant allele fraction for the variants from an AML 43 bone marrow aspirate sample. So you can see that most of the variants fall at about 40% or something. I just gave away the punchline. 40%, so they're present in all cells in this bone marrow biopsy. Blast percentage was about 80% or whatever. Some are at lower frequencies, they're subclones, but that's the AML 43 bone marrow sample at the AML 43 position. So we also sequenced AML 28, which is a totally different patient. We sequenced a bunch of different samples from this patient, including skin, remission sample, relapse sample, a bunch of others. And this bottom figure, I'm showing the same AML 43 positions in AML 28. So these shouldn't share, they should share maybe a couple somatic mutations that are recurrent, but that's it. Everything else should be absent from AML 28. And as you can see, the scales are different here, but the AML 43 variants are present at about 5% in this sample, and they should be not present at all. So this is an example of cross-contamination in this experiment, and we actually did a different experiment where it really mattered if the variants were in one sample or another, and we had to throw away the whole thing because it was completely convoluted. So this is kind of, I think, an example of a perfect storm of contamination that usually doesn't happen just because of the nature of the way the pools were created, but is an example, I think, of the level of contamination that can occur between samples when you pool them together. So this happened a couple years ago, and it raised a lot of questions. I wanted to know why, how this happened, at what stage. Obviously, it could happen in the laboratory stage. It could happen on the sequencer. I wanted to do some work to figure out what the parameters were that set up this sort of perfect storm of contamination. So one question is whether or not sequences were just improperly assigned to the wrong index. You thought it was from index 1, but it really was from index 2. So we did a little experiment where we took some human samples, and this was with our clinical assay. And for these, we actually spiked in M13 DNA, made libraries from it, and pooled it on the sequencer along with target-enriched samples from clinical specimens, and sequenced them together in multiplex, and then demultiplexed using our standard demultiplexing method, and then essentially just counted the M13 hits that were in the human samples. And so we observed 2 types of contaminants. So one, I'm calling the chimeric read, where one read in the read pair mapped to M13, the other mapped to the human genome, and then a non-chimeric read pair. So they both map to M13. And so on the bottom plot, I'm showing data from about 120 or so samples it's like 12 lanes of a high-seq instrument. And I'm showing these two types of contaminants together. Red is the chimeric and blue is the non-chimeric. And I'm showing, so that this is the contaminating reads and the percent of mapped. So in this experiment, contamination was pretty low. I mean, 0.0, it was the mean was 0.00545% across all these samples. Interestingly, and for most of them, the chimeric and the non-chimeric were roughly similar. However, for one lane, there was an order of magnitude higher non-chimeric read contamination. So it's still relatively low, but I mean this suggests that maybe, and I think that the non-chimeric contaminants could be laboratory contamination. So some carryover in a pipette or something. And so in general, this type of contamination seems to be low in our system, but you may have kind of a jackpot effect where some event in the laboratory could result in higher levels of contamination. So another question we had is, we tend to do single end indexing. So the library molecule, you just put the index on one end and you obtain that read. But recently, Illumina is moving to dual end indexing, and I think that's going to be standard for a lot of things. And so we wanted to know if that improves the crosstalk that can occur. So we designed a similar experiment. We did that. We mixed M13 with human libraries and pooled them together for sequencing, and we compared various demultiplexing strategies. So here I'm showing where we just considered one end of one index when we demultiplexed, and we considered no mismatches, and we considered one mismatch, so a less stringent demultiplexing strategy. And for this experiment, we actually observed even less crosstalk. So I'm showing here the scale has changed. I'm showing the number of contaminating reads per million mapped reads. So this is a low level of contamination, not much of a problem. But you can see that when you add the possibility of a mismatch in the index sequence, you get about twice the amount of contamination. And the chimeric and non-chimeric contaminants are about the same. When you consider both index sequences, you reduce this to basically zero. There's like 1 or 2 reads per million mapped reads. So dual indexing does seem to reduce the index crosstalk that happens either on the sequencer or at some other stage in the library prep. And so other experiments have been done. We're still kind of in the works to look at this sort of paradigm in a pooled capture setup, because, like I said, people do that for increased efficiency, and it is appealing because it can reduce costs, but not at the expense of massive contamination. And so we're looking into whether or not using dual indexes and this sort of thing can maybe make it possible to pool samples before you do the capture. And I've seen some data from the Genome Institute at WashU suggesting it does improve things, but I don't know if it's at the level of like a clinical test. But we're preparing some experiments to figure that out. So this leads to I think these experiments are nice, but it looks like, at least in this sort of experimental setup, contamination is pretty low, and it's not going to influence any sort of clinical calls. You're talking about a few reads per million. I mean, you're basically never going to see that when you are doing mutation identification, even in a somatic setting where you're looking at low frequency variants. But stuff happens in the lab. Samples do get mixed, and I think that some routine monitoring of contamination is a good idea. And the sequence data we obtain has a rich set of inherited variants. There's lots of structure in the human genome and human genetic variation, and we should be able to use that to identify cryptic admixture in samples, in data, after we've obtained them. And this obviously is not a new problem. People have published methods for doing this, and this is one of them. It's called the program is called Verify BAM ID. It was published by a computational group in 2012, and I think this was actually used by large centers like the Broad as a routine sort of contamination monitoring approach in their large sequencing studies. And it uses a maximum likelihood method for detecting mixed samples, and you essentially just pass it your sequence BAM file and then a list of known SNP positions, not that were identified in the sample, but just are known in the population. And essentially it goes down and it looks at those positions, and given the base counts at that position, it creates a likelihood of contamination and estimates a contamination parameter. So this has been, it was published and it's been used mainly on constitutional genomes and has not been applied to cancer samples, which may have skewed allele fractions in the sample. And that's essentially, that's the substrate that this program uses to identify contamination. So it's unclear how it would work on cancer samples, which is the main sample type that we sequence. And then another approach that I came up with was a haplotype-based approach to use closely spaced SNPs that are essentially within the length of a read, and to choose these SNPs that are essentially independent in the population. So if two SNPs are not independent, if they're completely dependent, then there's essentially only one or two haplotypes in the population. But if they're independent, then you have more than two haplotypes. And if these are common SNPs, then these haplotypes occur commonly in the population. And if you observe in a single individual, you only expect two haplotypes because it's a diploid organisms. If you detect more than two, then there's some level of contamination. And I think this could be a really good confirmatory test, because detecting three haplotypes is, there's very little laboratory artifact that can produce that talking about SNP pairs that are within 100 base pairs of each other. So this could be a good method for screening for contamination in samples. And so we've done a little work to test this method. So here I'm showing, so we have more mixed Hapmap samples, and I'm showing a plot here where on the x-axis is the mixed proportion of these two samples, and the y-axis is the contamination estimate from these two approaches, so verify BAM-ID and this multi-haplotype approach. And so the y equals x line is unity, and so the estimates look really pretty good. They fall pretty close to the line, suggesting that the estimated contamination from these programs is close to what we actually mixed together. And this is based on our clinical cancer panel, which is a few hundred kb. And there are about 200 of these informative SNP pairs that had enough coverage that you could actually analyze them. And in the mixed samples, there are at least 10 loci that were informative for detecting and estimating the contamination using the haplotype method. So that's kind of the level of information content that you get when two samples are mixed. And so, an important question is to know whether this works in cancer samples that have aneuploidies and other copy number changes. And so this is a very similar experiment, but instead we mixed, this is an in silico experiment where I took data from hopefully pure cancer samples and mixed them at different ratios and then estimated the contamination. So again, the two methods perform fairly well. They fall close to the line, suggesting that the estimate and the mixed proportion are very similar. But there were some pure cancer samples in this experiment, and the Verify BAM-ID estimated 40% contamination in these two samples. And I don't think there's 40% contamination in these samples. So an example of this algorithm does not account for copy number changes that can skew the allele frequencies. And this figure maybe comes up a little bit, but this is a variant allele frequency plot for the cases that came up positive. On the x-axis, I'm showing all the variants that were identified in this case. On the y-axis is the variant allele fraction. And most of these are SNPs, so they should be at 50% or 100%, and it's all over the place. And so it's samples like these that the Verify BAM-ID approach doesn't work very well for estimating contamination. So I think that these could be reasonable approaches for routine screening for contamination in clinical sequencing. And so I've written a little script that you can run on a BAM file that generates a report like this. At the top here, you see the contamination estimates using this various approach. So you get the Verify BAM-ID contamination estimate and some data from this haplotype approach. And you get a VAF density plot. And these are all the positions that you pass to Verify BAM-ID that it used to make its calculations. And again, if it's a pure sample, you should have a peak at 50% for heterozygous variants, a peak at 0, where there were no variants, and a peak at 100% for homozygous variants. This is an actual clinical case that we ran, which looks contaminated. However, it should be, because this is a bone marrow aspirate from an AML patient who was transplanted and had a relapse. And so 18% of the cells were positive for a rearrangement that was present at diagnosis, and our estimates were 16%. So, so this checks out. It doesn't appear to be a contaminated case, at least laboratory contamination. Okay, so just summarize this. So, contamination does happen, although, I showed you some scary data from the research side that used, pooled capture approaches, and I think that's really the biggest culprit. If you do single-end indexing and you pool capture, that's bad. You can have a high level of contamination there in the right scenarios. And I think this mainly happens because after capture, you do an amplification step, and you get incomplete extension of the products. One molecule is priming off another molecule, and you get chimeric species. We have experiments to try to test this, but I think pretty sure that's at least participating in that phenomenon. We did do these experiments with M13, which suggests that on the clinical side, index swapping, both in the laboratory and on the sequencer, are really rare, and I wouldn't expect them to influence clinical calls. But based on this experiment, I think that dual indexing probably does help reduce index crosstalk in the post-capture pooling stage. And then, the data we get from next-gen sequencing should allow you to routinely monitor for contamination. And I think two viable methods are this BAM-ID approach, which is fairly easy to implement, and then my haplotype method may be another approach, but I'm sure there are other and more sophisticated methods that could be available. And one thing that you could do is you could target specific loci that would be highly informative for this sort of thing, and it really wouldn't require extra space. If you were to go forward with this haplotype-based approach, I've looked through the entire genome and picked out hundreds of good candidates that seem to be from high complexity sequence regions that aren't paralogs and this sort of thing. And so you could easily target those with one probe each and implement that into your sort of quality assurance program. Okay, so now I'm going to shift gears to a little more of the research side, but I think that some of these, what I'm going to talk about, has some clinical applications. I've been doing postdoc research work with Tim Lay, and so, if those of you who are in the leukemia field know that he's been doing a lot of sequencing of AMLs over the last several years as part of the TCGA studies and his own work. And we really are getting a pretty clear picture, I think, of the somatic mutations that occur in your average AML specimen. So TCGA sequenced 200 AML genomes and published it last year. The Lay group and the Genome Institute sequenced many of these using whole genome sequencing. Others were sequenced with just exome sequencing. But this data set really, I think, captures the spectrum of mutations that you expect to see in leukemia. The paradigm that they've used for several years now has been whole genome sequencing of tumor normal pairs to identify candidate somatic mutations, and then design patient-specific capture reagents to target sequencing to those regions to confirm those variants. So they get high depth of coverage, and they can kind of cast a broad net initially, and then filter out the noise using this follow-up capture approach. So just to kind of foreshadow, one nice consequence of this is we have patient-specific reagents in the lab that we can use for lots of follow-up studies. And so I'm going to talk about that in a few minutes. So what we've learned from this is that the average number of coding mutations in an AML is about 10, so not that many. However, there are hundreds to over 1,000 passenger mutations, or presumably passenger mutations, that occur elsewhere in the genome. And these are really useful for tracking subclonal populations in AML. And really, it's difficult to identify subclones without these mutations, because you're only talking about 10 coding mutations that you would detect with like exome sequencing, for example. So, despite the fact that we've identified all these subclones, really we have no idea what they mean and if they're important. And so, that's going to be the focus of a lot of work in the near future, is to understand what the subclones mean clinically and if they have distinct functional properties. And that's the subject of what I'm going to talk about right now. Just to start, I want to illustrate kind of the paradigm that the Genome Institute and Tim and our lab has been doing for the past few years using a case example. So this is one of our favorite AMLs, AML 31. It's a 55-year-old man with monocytic leukemia. It's a normal karyotype, and at presentation, he had greater than 90% leukemia in his bone marrow. And this patient did relapse after a fairly long remission. When the initial sequencing was done, we did the whole genome sequencing and then the capture validation and identified about 120 variants in the initial experiment. And I'm showing them here in this VAF plot. So on the x-axis, I'm showing the tumor variant allele fraction or frequency. On the bottom panel, I'm showing the coverage of the variants. And on the top panel, I'm showing a density histogram of the variants. And you can see there are 3 peaks here which correspond to 3 clonal populations. So the one clonal population is about at 45% or so, 46%, and these are what you'd call the founding clone variants. These are present in every single AML cell. There are also 2 smaller populations. So these are present in a subset of the cells, so one at about 30% and another at about 10%. So one thing to point out is that even though the founding clone variants have the highest allele fraction, there's a subclone in here that actually is the most abundant cell in the sample. So if these subclonal variants are heterozygous, then about 70% of the cells harbor those variants. So just because it's a subclone doesn't mean that it's some minor population. It may be the most frequent population in the sample. So this is the picture we got sequencing just the bone marrow at presentation. However, this was part of a study to sequence de novo AML and relapse. And when doing that, we see a pretty remarkable picture of subclonal architecture changing with disease progression. So on this figure, I'm showing a scatter plot. On the x-axis, I'm showing the variant allele fraction at presentation. And on the y-axis, I'm showing the relapse sample. And so you see the black points represent these founding clone variants that are present in every cell. And the other colored points are subclonal variants. And you can see that the subclones that are present at about 35% and 10% at presentation are now completely absent at relapse. So they're no longer there. However, additional populations of variants appeared at relapse. So we have 2 populations here that are in the green population is in every single cell in the leukemia because the bone marrow is about 40% leukemia at relapse. And so the green points are about 20%, so those are heterozygous variants present in probably every single AML cell. So in this paper, it was published in Nature in 2012, showed that the clonal architecture changes between presentation and relapse. So the initial instinct is to ask whether or not this means that these subclones have some special properties that allow them to survive chemo, emerge after relapse, these sorts of questions. Do they have clinical correlates that may be useful? And so last year in the lab, Jeff Clow, who's a hematopathologist in pathology with me, we're 2 pathologists in a Department of Medicine lab, embarked on a study to understand the clonal architecture of AML using a variety of approaches coupled with this patient-specific capture reagent to understand if subclones have distinct functional properties. So we had a collection of 19 AML patients, and we performed this targeted sequencing on a variety of samples from these patients. So we had the de novo bone marrow aspirate, we had bulk peripheral blood, we flow sorted different populations out of the peripheral blood and analyzed those separately. And then some experiments I'm not going to talk about today, we cultured the AML cells in vitro, and we injected them into immunodeficient mice, and then sequenced what we got out at the end of those assays to see if the subclonal architecture changed after those experimental manipulations. So the 2 questions I'm going to talk about today are whether or not there's regional heterogeneity in AML. So we know in solid tumors, if you sequence different parts of a tumor, you can get different answers. Different variants are present depending on where you sample. And that question had not really been addressed in AML. People had done studies on particular mutations one at a time, but nobody had studied the full subclonal picture in different specimens. So this would tell you whether or not particular subclones have a propensity to mobilize to the peripheral blood or not. And then, there are other cells in a patient's blood and marrow besides just the leukemic blasts, and we wanted to know if these leukemic variants were present in these other cell populations. So to address the question of whether or not there's regional heterogeneity in AML, we did a pretty simple experiment. We took the de novo bone marrow aspirate and compared it to peripheral blood taken at presentation. So these are obtained on the very same day, just different specimens. And here I'm showing two AMLs, AML 31 and 28. I'm showing the variant allele fractions that we obtained from each specimen. And the diagonal line is unity, so if a point falls in that line, it was the same in both specimens. And you can see that in both of these cases, the subclonal architecture and the variants within those subclones are present in equal VAFs in both specimen types. There's no enrichment or depletion of subclones in the peripheral blood relative to the marrow. So if you plot all 19 cases on the same set of axes, you can see that this is really the rule and not the exception. The vast majority of variants, the vast majority of subclones really are equally represented in both specimen types. So the one interesting thing to note is that in the peripheral blood, the VAF of the founding clone is near 50%. So this means that, and this is bulk peripheral blood. So this means that every single cell in that patient's blood must be heterozygous for these variants. So as you know, peripheral blood may have some leukemia, but it's not all leukemia. So there are other cells in there. Do they have these leukemia-associated variants as well? And so we, from a subset of the 19 patients, we flow sorted out several different cell populations. We isolated lymphocytes, shown here, high 45, low side scatter, the blast population, monocytes, and then a maturing myelomonocytic population that includes myelocytes and neutrophils. And again, we flow sorted these, we isolated DNA from them and did capture experiments for the patient-specific somatic mutations to compare to the peripheral blood and the marrow. The results of that are shown here. And again, I'm overplotting all 19 cases, or all the cases that we did on one set of axes. And so on the left, I'm showing the blasts. And you can see that, as one might expect, the founding clone variants are present in basically all the blasts. Non-blast, non-lymphocyte cells also harbor all of the founding clone variants. You can see, however, that there are some populations that fall off the axis, suggesting that those subclonal populations seem to be enriched or depleted in those. And I'm going to talk about that more later. The other thing to note is that in lymphocytes, they generally lack the leukemia-associated mutations. Now, there is some low level here, which part of that is flow contamination. We did manual counts after sorting, and you do get some myeloid cells in after sorting, and there's debris and that sort of thing. So some of this is within the realm of flow contamination. But I should say that we also identified DNMT3 R882 at 10% VAF in the lymphocytes, which probably is not flow contamination. So there is some carryover, but in general, the leukemia-associated mutations and the precursor cells that have those mutations do not contribute uniformly to lymphopoiesis. So that's the main message here, is that most myeloid cells in the peripheral blood harbor the leukemia-associated variants. I think there's implications for the function of cells that harbor these variants. They have the capacity to differentiate, and based on the fact that different subclones are enriched in these populations, that maybe there's differential ability to differentiate in these populations. Lymphocytes generally do not have the variants, and then I just said the last part. So I think that this has implications for what these cells are actually doing. So if we zoom in on a few particular cases, and this is an AML 28. So I'm showing here the bone marrow variant allele fraction, and the blast variant allele fraction, and it's pretty much falling on the diagonal, suggesting that the subclones are equally represented in these two populations. But if you take this maturing myelomonocytic gate, you see a much different picture. So the subclone 1 and 2 here are depleted, so they're present in only a minority of these cells, where this rare population, subclone 4, is essentially not present at any appreciable fraction in the bone marrow. But when you sort for this population in the peripheral blood, and these are mature cells. Some of them are neutrophils. You greatly enrich for this particular subclone. So, I mean, is this subclone even leukemia? I think you could maybe make that argument, although in some labs that's heretical. But in AML 31, we see a similar picture but a different population. So on the left, I'm showing the blasts, and you can see that there's substantial skewing of this subclonal population. So subclone 1 and 2 seem to be depleted, and again, this rare population present in only the average VAF is 3% in the bone marrow, is greatly enriched in the blasts. And in the monocytes, you see this picture where it's falling on the diagonal. Okay, so just to recap that part of the talk, so this study, I think, showed in this set of 19 AMLs, which is fairly representative of most AMLs, it did not have some of the canonical transcription factor fusions that are recurrent fusions that are common in AML. So that's one difference that could affect these results. But in this set, the peripheral blood and bone marrow show essentially the same subclonal patterns. And I think that this has implications for molecular testing, at least at diagnosis. Now, for MRD type monitoring, bone marrow still may be the preferred specimen, but in diagnosis, you may not need bone marrow to do routine molecular testing. In that presentation, in an AML patient, virtually all myeloid cells appear to be derived from the leukemic clone. And I think that has really significant biological implications for the heterogeneity of the hematopoietic stem cell compartment that gives rise to AML. There may be multiple cells, some of which comprise the majority of the leukemia, but others may be pre-leukemic and destined for relapse if they're not eradicated. It also means that AML, although defined essentially as a differentiation block and accumulation of immature cells, this differentiation block is not complete. Based on our study, the lymphocytes are not part of the leukemic clone, at least uniformly, or the cells that have the leukemia-associated variants are in progenitors that generally do not give rise to lymphocytes or are skewed towards to the myeloid lineage. And then in some cases, and I don't want to, I mean, this is not uniform, but in some cases, subclones are enriched in certain cellular populations. And again, I think that this means that there's evidence for differential function of different subclones and evidence for heterogeneity within some progenitor compartment that contains the leukemic cells. So, a big question, I've talked to people about this today, is what happens to subclonal variants over time. And this is a question that's really going to take longitudinal sequencing of AML patients, starting at presentation, after chemo, and maybe periodically throughout the course of their disease. And so at WashU, we're kind of starting to ramp up this enterprise. And the assay that we're using is what we're calling an enhanced exome. It's using a Nimblegen exome reagent. And I think a pretty neat trick that could have implications for clinical testing is we spike in probes targeting the genes that we really care about to improve the coverage at those critical genes. And so I think this is kind of a general, could be a general tool for sequencing when you want to sequence the whole exome, but you really care about a few genes that need to be, say, clinically reported, because then you're not going to miss things in those regions, but you do get the rest of the exome. This is ultimately going to be a validated assay performed in a CAP-CLIA environment. That work is in progress. And the design at this point seems like it changes a lot, but we're going to be doing tumor normal pairs with a day 0 marrow, and then we're going to sequence at day 30 after chemo. And the point here is to track what happens to the leukemia-associated variants after treatment. And we want to know, does the disease clear? Do some variants or some subclones persist that at some later stage we could do a retrospective study and determine if that has any sort of prognostic value. And the study is designed so that we're going to track clinical outcomes and what changes in treatment happen as a result of these genomic studies. So I think this is another thing that really needs to be addressed is if you apply sort of Cadillac genomic testing to a disease, does that really make any difference? Because we already do a lot of molecular testing for leukemia in particular. And so when you do it all, does it really help? And that question is going to take longer than just a couple of years to answer for sure. So just kind of a quick sort of anecdote. We've applied this paradigm to just a few cases, kind of just in an initial sort of discovery phase. And so I'm going to show some data on 2 patients. The day 30 pathology in both was morphologically negative. The outcome was drastically different. One of them relapsed at day 90, and the other was in complete remission after 673 days. And so I'm showing, this is a plot of both patients. So that the top patient is the early relapse patient. I'm showing the day 0 VAFs. So this is exome sequencing, so we don't get that many variants. I'm showing the ones here, and they're highlighting some of the recurrent mutations that were, or genes that are recurrently mutated in AML that were identified in this particular patient. The bottom case is the one with the long remission. So both have some collection of variants. And when you sequence the day 30 marrow, the patient that relapsed at 90 days still had detectable disease by this assay. So the cluster of variants with a mean VAF of around 10%, suggesting that 20% of the cells in the sample are heterozygous for these variants. Now, the patient didn't have an excess blast in their marrow, so I don't know what these cells are. They're not blasts. The long remission case essentially was undetectable for the variants identified at day 0. So, this is the kind of question that we and others would like to ask is, this assay is not an MRD assay, but some sort of longitudinal tracking of variants in these patients could be clinically useful to decide whether or not patients who wouldn't get a transplant, may be candidates, and to understand, what the natural history of their disease is. Okay, so that's all I have. I just want to thank a few people. The Department of Pathology, it's kind of grown into a big operation, but I've worked closely with Eric Dunkevich for a long time on a lot of these projects. He's an assistant professor there, and he's a really great guy and very knowledgeable about next-gen sequencing. In the lay lab, thank Tim's, who's an excellent mentor and very enthusiastic guy. And Jeff Clow is a hematopathologist who sits right next to me, and he's a very smart guy. He's leaving to go to St. Jude in about 5 weeks, and so we don't have much time together left, but he's a really great guy. And the Genome Institute, which does all the AML sequencing, and particularly Chris Miller, who kind of leads the analysis group there and provides us with the data. And that's it. Happy to take any questions.
You said you're using tumor normal pairs. What are you using for normal?
So that is an excellent question. We've done extensive work on that. The question was, the question was for the tumor normal pairs for our longitudinal sequencing, what are we using as normal? And that's a great question. That's been in debate for a long time, and that's actually how our sorted blood population study kind of got started, because Tim was adamant that lymphocytes would not be the best normal because they could contain some variants and so forth. Turns out they don't. But the choice of normal is important because in day 0, you can have a lot of skin contamination with leukemic cells, which can really make it difficult to identify the somatic variants. In fact, MLL leukemias in the TCGA study have fewer somatic variants than other cases. They tend to be monocytic, and they tend to have high white counts, and they tend to stick in the skin. So that phenomenon could just be an artifact of lots of skin contamination. So we've tried mouthwash. Day 0 mouthwash has the same problem, and you sequence a lot of bacteria too, so it doesn't work very well. Our current paradigm is to take day 0 skin as a backup, take day 30 skin, and use that. I think we're actually going to take day 30 mouthwash as well. And if patients die before day 30, then we'll have to use the day 0 skin, but that's the current plan. But it is a crucial question because the skin contamination can essentially make it impossible to identify somatic variants in a discovery basis.
So I'm kind of thinking about this like a microbiologist rather than an oncologist, but it seems to me if on day 30 you have the emergence of this new subclone, you would say this is somehow resistant, but you don't, or resistant to the therapy,
mm-hmm.
right? But there's no way to flow sort prospectively because you have DNA-based things. So could you, like, singly clone by limiting dilution these cells and then do your sequencing on both the patient and then find those to see if the certain subclones do actually have resistance to the chemotherapy?
Yeah. So, yeah. Maybe I can just repeat a new question that I'm not sure if I totally understood. I mean, what you're saying. I think that if we could sequence single cells, and people do this, that would be interesting. But to prospect, so the idea would be, I think the question is, can you flow sort single cells at day 0, sequence those single cells, identify the complicated subclonal architecture in those, and then use what you identify at day 30 to no.
No, I mean, so you can take lymphocytes and you can plate them one per well, and then they'll grow. You can grow them up, so you have lots, right?
Yeah.
So that's not. But if you have a subclone that emerges in your whole gross pool, the question is, like, could you find it then in your plate of cells and ascribe to it some behavior that was different than the founder that then has a clinical correlate?
Yeah. Okay. So the question is, can you, in the lab, in vitro, culture primary AML cells, see what emerges there, and maybe what responds to some treatment, and then put that in the context of what happened to the patient after they've received chemo, say. Is that kind of close enough? Okay. So we can culture primary AML cells, but not well. They expand maybe 1 to 2-fold over the course of 2 or 3 weeks, and then they die. So that those experiments I didn't talk about. So Jeff Clow, when he joined the lab a few years before me, developed a system to do this culturing, and we, in our paper, we cultured several of these primary AMLs, and in fact, this AML31 with the blast clone, that clone grows out of this culture, and in 7 days, it goes from 3% to like 20% or something. That happens to be the subclone that's present at relapse. I think that's just a coincidence. Now, if we could do this on a large scale and see what subclones grow out in culture, do they correlate with relapse? I mean, we can do that. In our little study, we tried, and there's no correlation. We also put the cells into a mouse and see which subclone grows out of the mouse, and does that correlate with relapse? People have done that, and they've said various things that it does, and the subclones that engraft do correlate with relapse. We didn't observe that in our study, although it was underpowered. I definitely think that. So what I would like to do is I'd like to flow sort. This would be incredibly hard to do, but if you take some sorted progenitor population from the so you sort hematopoietic stem cells from an AML sample that has this complex clonal architecture which we've characterized, and then if you could separate those out and study those just from the de novo sample using by sequencing and by epigenetic studies, like that would be really interesting to see if there are differences, functional differences in the HSC compartment that correlate with subclones. I think that would be really interesting.